Everyone, my name is Rawan aldubaian. I am a PhD student at UCL in cell and gene therapy. And today I'd like to present a research on the impact of reducing dimethyl sulfoxide, or better known as DMSO, concentration from 10% to 5%, and its effect on the viability of CD34 hematopoietic stem cells after cryopreservation. Now, this study was conducted at the King Faisal Specialist Hospital and Research Center in Riyadh, Saudi Arabia. Now, just to give a quick background about DMSO, it's a commonly used cryoprotectant for storing hematopoietic stem cells. However, high concentrations of DMSO can be toxic to cells and can lead to, as I was saying, that DMSO is commonly used cryoprotectant for storing DMSOs, for storing hematopoietic stem cells. However, using high concentrations of DMSO can be toxic both to cells and lead to adverse reactions in patients, either during or after transplantations. Now, the standard concentration used in our institution was 10% before changing it to 5%. And the reason we decreased the percentage was because during the COVID-19 pandemic, we increased the number of cryopreserved stem cells. And it was for two main reasons. One of them was to ensure we had sufficient doses for transplantations. And the other one is that we wanted to prolong the window between the harvest and the infusion. And as a result, this caused higher incidence of adverse reactions that were linked to DMSO toxicity, especially in pediatric patients. And in order to address this issue, we decided to initiate a study to investigate whether reducing DMSO concentrations could maintain cell viability as effectively as the standard 10%, especially that other scientists and researchers worldwide have started to explore using other substitutes to DMSO or trying to test lower concentrations. And we can see that with the CAR T cell therapy. You can see that some companies use 5% already, or the 7.5%. So we decided to test that with hematopoietic stem cells as well. And the goal was to explore the potential benefits of using lower DMSO concentrations in terms of reducing adverse reactions and improving patient safety. And if you can see here in the method, so what we did is that we obtained fresh hematopoietic stem cells product from healthy consented donors, and we divided the product into two groups, right? One was frozen with 10% DMSO, and we used this as our control. And the other group was frozen in 5% DMSO, which was our tested, at the concentration we want to test. And before freezing the product, we measured the viability of the cells using flow cytometry testing to use this as our baseline to compare with the post-thaw cryopreserved product. And so then when we took each aliquot from each group, the 10% and the 5% DMSO, and we thawed them at different time points, being 2 weeks, 4 weeks, and 8 weeks. And we measured each one of the samples using flow cytometry analysis again to test to measure the viability and the cell count of stem cells using the CD34 markers, and then we compared them to the fresh product pre-freezing. And so you can see here in the results of the study that our analysis revealed that there was no statistically significant difference in cell viability between the 10% and the 5% DMSO groups, which was a great thing. Moreover, here, if you can see the CD34 levels, you can also see no statistically significant difference in CD34 levels or count even though like the 5% DMSO groups consistently showed higher CD34 levels in comparison to the 10% DMSO across all time points, it still wouldn't be like as a statistically significant difference. So if the viability of stem cells stored in 5% DMSO is comparable to those stored in 10% DMSO, it could actually support the use of lower DMSO concentrations, which can minimize adverse reactions. And this is fantastic. And it's particularly important for pediatric patients who are more susceptible to DMSO toxicity. Furthermore, like another advantage of using a lower DMSO concentrations was, so the current calculation for DMSO dosage is based on the body weight, right? So each patient can take 1 ml of DMSO per 1 kg. So, and this sometimes can limit the amount of stem cells that patients, especially with lower body weights, can receive. And as a result, this can lead to prolonged or unsuccessful engraftment because they would have 2 day zeros instead of 1. And then by using a lower DMSO concentrations, we can actually ensure that patients receive the full dose of stem cells without the risk of DMSO toxicity. So in conclusion, our study suggests that reducing DMSO concentration from 10% to 5% does not actually compromise the viability of hematopoietic stem cells. In fact, it may even lead to higher CD34 levels. And this finding is amazing because it's important. It has important implications for improving patient safety and optimizing stem cell transplantation procedures, especially in pediatric population. Thank you very much for your attention.